सो मॉर्गन ने जो है 2 एक्सपेरिमेंट किए थे. मॉर्गन सर हैव डन 2 experiments. So we are going to take an overview of both the experiments, that how linkage and recombination actually works. So there is one cross that is known as cross A, and there is another cross known as cross B. So there are different kind of characters which are taken into consideration in both the crosses. So in one cross, the parental characters are yellow and white. We have talked about it in this lecture, that yellow body and white eye are the characters, and the second one is going to be of wild type. Okay, and in second one we have taken white and miniature. Okay, miniature wings के बारे में हमने यहाँ पे बात की है कि white और miniature को हमने यहाँ पर माना है अपना second character. First character is white and second character is miniature, and the second organism taken is of wild type. So इनके बारे में अब cross होगा इनका. Okay, so there is cross A which is having 2 characters, yellow body and white eye, that is the body color and the eye color. The second individual is of wild type, and in second cross the 2 different characters are taken, that is white and miniature. So in the very first character you can observe that there are 2 genes, that is small y and small w, and these are present very close to each other. These are present very close to each other in a fine vicinity. But when we talk about small w and small m, these are present quite far. You can have a look at both the images. In this particular image these are present quite close to each other. In this particular image the w and m are quite far. It means that the linkage is more in cross A, that is the 2 characters, that is the body color and eye color, are more linked, and the second thing is not that much linked. So linkage is more in cross A, linkage is less in cross B, and we have talked about the linkage and recombination and the relation between them. So this is going to have very लो फ्रीक्वेंसी ऑफ रिकम्बिनेशन, क्योंकि यहाँ पर लिंकेज ज्यादा है, एंड लिंकेज एंड रिकम्बिनेशन आर इनवर्सली प्रोपोर्शनल. सो मोर द लिंकेज लेस विल बी द रिकम्बिनेशन, लेस विल बी द लिंकेज मोर विल बी द रिकम्बिनेशन. सो जब इनका क्रॉस होता है so we will make a cross and an F1 generation will be produced. So in that F1 generation the wild type and yellow white type organisms are produced, and the parental type are 98.7, means जितने parental type थे 98.7 are present as it is in the next generation. It means that the linkage was very high. Since approximately 99% of the individuals are exactly similar as the parents, there is no change. आप समझ रहे हैं, linkage बहुत ज़्यादा थी, so the genes present in the same chromosome are transferred as it is to the next generation, and there is no change in them. That's why the parental type combinations are 98.7%. And the recombinant types are very few, that is 1.3%. This is because the linkage is quite high, and since the linkage is high the recombination is low. In the second case there is a little bit low linkage because the genes are present quite far from each other. That's why in F1 generation the parental type individuals are 62.8, which is quite less as compared to this one. So we can conclude that Y and W are more linked as compared to W and M. Okay, so there will be more recombination in W and M as compared to Y and W. So here the recombinant type individuals are more as compared to the first cross. Why? Because there the linkage is less. अगर linkage less है तो recombination ज़्यादा होगी. And what is the number of recombinant types? 37.2. It means that the parental type are only 62.8 and the recombination frequency is very high in this case, that is 37.2. In the last case it was just 1.3%. So there is a huge difference. And when we will intercross the progeny is formed in the F1 generation, तो हमें F2 generation मिलेगी, and that F2 generation will not be resembling with the data of dihybrid cross that is done by the Mendel, because the ratio of Mendel's dihybrid cross, the phenotypic ratio was 9:3:3:1, but ऐसा कोई data हमारे मॉर्गन साहब के पास नहीं आया. So the data of dihybrid cross done by Morgan was very different from that of Mendel. Okay, that is because the linkage, because of the linkage, because more is the linkage. For example, in this case more is the linkage, less will be the recombination, and less will be the recombination, the recombinant type were very few, and in this case the linkage was less, so the recombination is more. That's why the recombinant type of individuals or the organisms in that progeny were 37.2%. This is the concept of linkage and recombination, that more is the linkage, more will be the genes linked together with high अगर एसोसिएशन में बहुत ज़्यादा है तो उनमें क्या होगा, फिर रिकम्बिनेशन कम होगी. लेकिन अगर लिंकेज कम है तो क्या होगी, रिकम्बिनेशन ज्यादा होगी। That is about the linkage and recombination. I hope you like the video. If you really like the video, then hit like button. And if you are watching my video for the very first time and you found it beneficial, then share this video with your friends and tell them to subscribe my channel too. Thank you so much and stay tuned for the upcoming videos on chromosomal theory of inheritance. उसके बाद पढ़ने की कोशिश करेंगे about genetic maps as well as different process which we have skipped. That is complementary genes, supplementary genes, dominant recessive epistasis, polygenic inheritance, and all other things which are very very important from examination point of view, and some important videos on pedigree analysis too, so that you can solve any kind of pedigree given in the examination. Thank you so much.